Today, we're going to talk about some newly published apps on BDC. So my name is Dave Robertson. I work for a company called Valsera, but I'm the community engagement manager for Biodata Catalyst powered by 7bridges. So we're going to learn about some new features, and these have all been documented in the release notes. Now, this is something I don't think everyone checks. You can find the release notes on Gitbook. So here's the Gitbook link right there. What else? So we're going to focus on this year, 2023, and we have there were some release notes in January, and then they're pretty much every month. And I just plucked out some things that you can run on 7bridges, but this is not everything on the release notes. These are just 3 things that I wanted to share that we haven't talked about before. There's a lot of great information on the release notes, such as the BDC picture API, the new API. Even more importantly, the data releases are in there as well. So it's a great resource. I would encourage everyone to check it out. So yeah, and the STAAR pipeline, which we're going to talk about first, has not actually made it to a release note yet, but it is available on Biodata Catalyst. So we're talking about apps in particular, and I just wanted to sort of define that word a little bit more, because I think apps are core to our platforms and our community. So being the 7bridges engagement manager, I'm going to focus on 7bridges today, but you can run apps on Terra as well. On 7bridges, there are really 2 categories. So there's CWL description of workflows or tools. So that's one category. Second category is interactive web applications. That's something I've never demoed, but we'll get into that. So these are fully interactive websites that you can get immediate feedback from. So what are the benefits of both of these? So they're easy to run via GUI interactively, or even via an API, like you can embed it in another script. They're scalable, they're shareable, but most importantly, I think they're accessible to a variety of users, clinicians, biologists, etc. So how do you find apps? So if you log on to Biodata Catalyst powered by 7bridges, in the main sort of drop-down menu, there's public resources. So under that, you can see datasets, files, but and this is slightly changed a few months ago than it was before that. So workflows and tools are where you'll find the public apps gallery, essentially. Interactive web apps, you can click there. There's only a few so far, but we want to collaborate with the community to make more. Projects are, of course, the public projects, which are example analysis. That's been fully sort of tested for you and a great way to learn. So, but I want to mention this, and I don't think we do this enough. Really, the power of Biodata Catalyst is in the connections. So today we're talking really the work of 3 scientists. Dr. Lee at Harvard, so he, with his collaborators, also developed the STAAR Pipeline. Dr. Hugh at UCSF, MetaCyto, and Dr. Hugh at NCI CBIT, OmicsCircos. So sort of by coming today and by participating in the community, you're also connected to these scientists and the rest of their teams. So let's start with the STAAR pipeline for GWAS. So let me pull up some notes. So this is new for me. I've never done a tutorial on this before. So bear with me, but it just came out for Biodata Catalyst powered by 7bridges. It was also available on Terra, which I'll get into. STAAR Pipeline stands for Single Trait Association Analysis with Relatedness Pipeline. So it's a new application designed to analyze whole genome or whole exome data, and it provides a really accurate representation of genetic data by accounting for relatedness within the samples, like some other pipelines do as well. And of course, it's great because it provides great association results and reduce false positives. So it's based on an R package. Great question, Prithvi. So I see it's coming in. So how was the relatedness calculated? So I don't know the answer to that. I'll have to get back to you. I think I'm assuming that you can use the same, like if you're going to use this on TopMed, I'm assuming that you would use the same relatedness matrices and PCA, but I don't know. Good question. So I think the package in particular includes two key features. So you can analyze single variants or what they call variant set analysis. So the single variant analysis gives us the capability to evaluate individual variants and get p-values based on things like minor allele frequency, of course, and minor allele cutoff frequency as well as other things. So going on, so if we dive deeper into what they call the variant set analysis, it consists of gene-centric versus non-gene-centric analysis of rare variants. Gene-centric coding analysis offers several genetic categories: putative loss, function missense, disruptive missense, combination of putative loss of function and disruptive missense, and synonymous. On the other hand, you can run gene-centric non-coding analysis, which provides even more categories. Thank you, Kritika. So this is the kind of collaborative stuff I want to encourage at our community hours and at other events too. So for non-centric gene-centric analysis, the STAAR pipeline includes features like sliding window, analysis with fixed sizes or dynamic sizes. It also has in-depth analytical follow-up of really dissecting the association signals independent of known variants, and that's through the feature called the STAAR Pipeline Summary. So that was the overview, and of course, use cases. Everyone knows this. This is right in our wheelhouse. These are widely used in genomics and medicine. Genotype, phenotype solutions are popular, and STAAR is becoming more and more popular. And I think it's unique because it has a unique way of enabling the single variant and the variant set analysis features. Okay, so how is it implemented on BDC? So the initial description was in the workflow description language, which of course works well on BD Catalyst powered by Terra. So it was published in 2022. And actually the lead author is Dr. Gaynor, who was a BDC fellow cohort 1, and I see Kenny Westerman as well. Anyone else? Who also was a fellow cohort 1. And this is a sort of a picture from their publication here. So, it's the same general procedure. You fit a null model, then you run what's called RV tests, and then you compile your results. What's new is the common workflow language description on 7 bridges, which runs very well on BD Catalyst powered by 7 bridges. So that's new. So what else do we have? So how would you use this? So you can copy apps from the public. It's in the public apps gallery. So here's the link, bit.ly.bdc-apps. I always like to promote this link in particular because we don't actually have to log in. So why don't I go take a look over here? So as you can see, I'm not actually logged in. So this is publicly available to anyone on the platform. So anyone on the internet, essentially. You don't have to log in. So these are all the CWL apps. In this case, it's really easy just to search for STAAR. And what I want to call out is that, so there's multiple tools here. The main one is the STAAR Pipeline, and you have a couple things I want to call out. You have the pipeline version. So as soon as a new version is released, this will get updated. And you have something, this is new, and this is really in my wheelhouse in particular. There's an effort to get these apps more portable. And so there's something called the CWL tool, which is a reference runner for the CWL tool specification. So you can actually run this pipeline anywhere that you can install CWL tool. So if you want to work on your local laptop or local HPC, you can run this description of the STAAR pipeline, as long as it has this tag here, CWL tool tested. So this is the main one. The other ones come into play as well. Something else about the CWL specification is that all this information is it's like a sort of self-describing file. So all this information about how to run the pipeline, to run the tool is here. And what's interesting here is you would run this multiple times. So as you know, in Genesis, there's a separate tool for the null model. Here, you're going to run this once to fit the null model. And then here are the requirements for the null model. So there's a question about the kinship. So it's sparse, of course. Kritika, you have a question. Go ahead.
Hi, David. Yeah, I have a quick question. I realized that you have to run a different null model. So does that mean the one that we have from Genesis is not compatible with STAAR here?
Great question. I'm not sure. I haven't had time to dive deeply into this, but I think we should check that out. I think we should meet and try to run it.
Okay.
But I'm not sure.
I got that question mark down, and let's find out, and we can follow up.
Yeah, I'm just specifically about the CWL wrapper around it. Instead of being like a workflow with multiple apps, you would run this once for the null model, get the results, and then run it again to do your association. So it is, I mean, the null model for Genesis is absolutely in our object. But yeah, I'm not sure. That'd be good to test. So it has common use cases, some important notes, and then of course performance benchmarking. So if you're if you want to know how much this would cost to run, I think these values and these prices are pretty good, because for instance, if we want to use even the largest chromosome 1, 61,000 subjects, I mean, it's coming in at $14, I think. Yeah. Which I think is pretty reasonable for that level of subjects, for that number of subjects, and also the hour. It's only running in 2 hours. So of course, you know, if you're testing, just use chromosome 22 or something like that, and it'll finish even faster and be much cheaper. So you'll see. Okay, so this is the general procedure to do the testing with chromosome 1, 10, and 21. So it's only 33 minutes for chromosome 21 for 61,000 subjects. That's of course the single variant test type, but there's several other types. So it's a really complex package, all in this one sort of object here, the STAAR pipeline. Okay. Any other questions about STAAR before we move on to MetaCyto?
From where you have shown the documentation, can you please say it again and show it again?
Sure. This one?
Yeah, this one.
Okay. Yeah, it's actually, I'll zoom out here. It's right here. So here, I'll put it in the chat. Everyone click on that. It'll should open right up. No login required. And by the way, if you ever, so there's more links here, source code, homepage, some deeper documentation as well. Hi, Kritika. Is your hand up again, or was that from before?
Yes, it's up again. So I have another question. I know you showed us the main STAAR pipeline. What are the other STAAR apps that you have in there? I mean, I was wondering if this is at all something that can be used on structural variants too. I mean, I did see something in Dwar, so I wasn't sure what that was. The second one here, STAAR pipeline.
Yeah, I'm not sure. That's another great question. I didn't do enough homework to understand what this tool is actually doing. I don't think it can handle I think it's mostly genotypes and phenotypes with annotations.
Okay.
So I don't think it, my understanding is I don't think it handles any other types of variant information outside of genotypes in the annotation.
Okay. And this is available already for use, right? The
Yep.
first analysis pipeline, that is. Okay.
Yeah.
The
So all
first
those,
app.
yeah, all these apps are available to
Okay,
use.
perfect.
And
Perfect.
this annotator is how you would create, I think what they're calling a special GDS format, which has the annotation. So it's something's very similar to the Genesis workflow, but there's
Yeah.
enough of a difference to, I would go slow, and we should probably develop some more learning materials around how to use this.
I would be more than interested in seeing, like, because you also have GDS file formats that are required for Genesis, and also your GRMs and what do you say, what was the first one? Yeah, the null model and all of that. If they are compatible, so you don't have to do 2 of those to run STAAR and to run Genesis. So that would be of immense help if we could get some clarity.
That's a fantastic question. And by the way, Kruthika, so something that was, I know you wanted to see a demonstration of Annotation Explorer, which is entirely based in entirely for Genesis. It wasn't working, so I'll just say it's all working now. There was an issue with public projects, cloning of public projects, all completely fixed now. So we can definitely meet, and I can show you a demo of Annotation Explorer.
Perfect. Perfect. Thank you, Dave.
So let's move on and talk about MetaCyto, which is something completely different and also even newer to, I think, this whole community. So it's a powerful tool for doing meta-analysis of flow cytometry and mass cytometry data. So it's a workflow designed to facilitate meta-analysis based on an R package again. So it allows researchers to jointly analyze cytometry data from different studies that may have diverse sets of markers. So the ability to perform a meta-analysis is significant in the cytometry field as it broadens the scope of potential studies, enabling integration and comparison of data types across multiple studies. So it's capable of identifying and categorizing cell populations based on marker expression and can compute population cell frequencies and median marker intensities. So I saw some polls going out, and I'd be interested to see, how many people have done cytometry analysis or have access to this data, because I think for this community in particular, it's very, very new. So the next slide, I had to do a little bit of homework to understand the difference here. So flow cytometry is laser based, so using the fluorophores, and can analyze cells in a mixed population. It's widely used in immunology, as an example. Mass cytometry, on the other hand, or cytometry by time of flight, uses heavy metal ion labels. And so my understanding is a higher throughput. So there's a greater number of parameters that can be measured simultaneously. So use cases. So I'm not aware of any use cases on Biodata Catalyst yet, and there's no cytometry data that's been hosted by NHLBI yet. Although I've heard that this could change very soon. But of course, like anything, it can be brought in via a bring your own data or BYOD approach. If there's any data that you have access to is publicly available, you can link it. There's multiple BYOD methods to do that. So this app was actually tested using import data sets, which is available on the internet. I think you do have to register, but I believe it's freely available. And that includes cytometry data on that website. So how is this implemented on BDC? So like everything, it's well, like the STAAR, it's available in the public apps gallery. So I'll just click over here. The way I find it is I go to the toolkit in this case and click on MetaCyto. So you'll see those apps there. I put it in the chat. So what's interesting, this was a workflow was created. So the other one is designed, so STAAR was designed to run sort of like a quote unquote pipeline as one tool. This one was implemented as multiple steps. It's still the same R package, but in this case, it was broken out into multiple steps to make it more extensible. And I like that in some cases. In this case, I like it because it's not that complex. So this is about the limit. If there's more complexity than this, it's sort of better just to leave it as a tool because it doesn't enable that much that many changes by the end user. So here's another view of the pipeline. So it's the first tool here. You import the data, you pre-process it, you identify subsets, and then you run the meta-analysis. So why is a workflow good in this case? Well, you can make use of memoization in your project, for example, where you could run the workflow, and then if you wanted to iterate, identify different subsets or change parameters for the meta-analysis, you wouldn't have to recompute these first three. Your project would remember that. You could also look at the view stats and logs and see specific computational requirements for each of the steps. So I think it's fine in this case, but I've seen some which are like 10 or 20 apps, and I think that gets a little harder to understand and work with. Okay, so this is something new, and I kind of want to get your feedback on. So meta-analysis for cytometry data from import data repository. So I encourage everyone to look at this link when you have a chance. It'll be in the slides in our forum. So FYI, so this is not on Biodata Catalyst. It's a link to another sister platform of 7 Bridges called Cavatica. But we can enable something like this, or basically import something like this to our Gitbook documentation. And it's a walkthrough of a public project. So it's not on BDC, but all actions that, make a project, add your apps, everything will be described. Everything described there could be done on BDC. So I just want to get some feedback when you have a chance about, is this cytometry sort of project something that's interesting? What I think is great is that it takes the public project, which you can actually clone now that they fixed it. It takes a public project and walks you through it, and then also provides homework assignments. So we can easily implement this in a workshop, a little mini workshop, if you guys want. So has anyone used cytometry that wants to share? Don't be shy. I have not. Yeah, I'm with you, Prithvi. I don't really know much about it, but I'm interested in learning more. So the next topic is OmicsCircos for genomic visualization. So what is that? So it's a visualization tool, so used in genomic research to analyze genomic data in circular format, like shown here on the right. I used to see it all the time in TCGA papers. It's used a lot in genomics, but it can be used for other types of data as well. So it's based on a bioconductor. It is a bioconductor package. So it shows the whole genome at once. So usually the main sections are chromosomes going around the circle. And then I think one of the key things is you have these lines, these links in between parts of one chromosome to another chromosome, so you can see fusion events. You can really pick those out easily. Of course, you want to filter down for you want to massage the data to see like the most significant events. So you could have fusions in the middle. You could have heat maps for mRNA expression. You can have GWAS data where you have sort of multiple circles of Manhattan plots, because Manhattan plot is by chromosome, right? So you could do that as well. Cytometry, does it relate to cytometry data? I'm not sure, but that would be cool to try. But of course, it's all about multi-omics. So genomics, transcriptomics, proteomics, etc. This was developed by Dr. Mirzman's lab at NCI. So some use cases. So this is not actually OmicsCircos. It's from a different R package, but I thought it was cool that if you do know some R code, you can make some really interesting things like a dartboard or some of these other things. So they get pretty complex. So it's kind of like you have to hold yourself back and not put too much data, because there's only so much data like the human eye can read at a time. But I think it's, especially for structural variants, I think it's, copy number variation, fusion events, super important there. So how did we implement it? So the implementation is unique here. So it's implemented as a interactive web application. So there's bioconductor. So what we could have done is, create CWL or WDL apps, right, to run this bioconductor package. But in this case, we made a R-Shiny application, and so that's hosted on Biodata Catalyst. So it would enable pure biologists to create these plots. And so this was also a collaboration with the Mirzman lab at NCI. Oops. So I wanted to demo this for you. So let me show you how to get, so I'm not even logged in. So let me log in on another screen.
If there are any other questions, just a reminder to, you can put them in the chat, use the raise your hand feature, or if anybody has a quick one now, we could probably address it while Dave's logging in. So don't be shy with questions.
So this is my dashboard. So remember, everything I talked about today is under the public resources drop down. So you have workflows and tools, which are the CWL apps, and then you have something new called interactive web apps. So if you click on that, there's 3 currently, but we want to keep adding more. So there's a couple for, well, there's one for Genesis, there's Locus Zoom, and the new one is the OmicsCircos app. So what you do is you click open, and it'll open up in a new window, which I'm going to put over here for you. It looks like this. So here, oops, let me make this better. So it really opens up in a new web page or a new tab, but it's still behind the NHLBI BDC firewall, so you have to be logged in to see it. It's not public, but it has. So this is the difference. So this is an interactive way to do your analysis, and it works very well for sort of things at the end of your analysis, like particularly visualization, because the data is already sort of reduced in size, and you kind of want to fine tune things. So of course you have a user guide here and references. So if you click Start Discovery, it could be my voice, I don't know. How am I coming through, guys? Is it okay?
It's okay on my end.
Okay, yeah, that could be my microphone. So this is how you would walk through the pipeline. So the Shiny app. So what's interesting here is that you have this toggle button here. So it has built-in data that you can use to play with, which I think is a great feature. Of course, you can use your own the data from the platform as well. So Genesis tutorial. It's probably going to complain because I'm not logged in. So great question, Prithvi. So he asked what kind of data should be given. I've never actually used it with GWAS data, but I like to try. This example is going to walk you through there. Let me refresh this. I think I'm logged out. This example in particular is going to walk you through mRNA fusions, basically omics data. So if you want to use the built-in package data, just toggle that. And so what example data? So it's select example data sets from the omics package. So what you want to focus on is up at the top, what sort of tab you're at. So segmentation, mapping, links, and then link polygon data. So segmentation would be, how do I describe this? So I think the chromosomes essentially. So it has HD18, HD19. So you just have to be careful. You can use you can use anything. I suppose there's you could use version 38 as well if you had that file in your project. So these are just the example data sets. So you click load, and then so what do you want to map? Once again, I'm using the built-in data as well. So for mapping data, we can use like the gene expression. So load those up. I think I'm doing this right. I'm a little out of practice, guys. So the link data would be fusions for sure. Link polygon data. So by the way, these are all TCGA, Cancer Genome Atlas open data sets. All right, let's see if I did this right. So, oops, let me zoom out. Okay, so we have our input data. At least we hope we do. And then we go to visualizations. So you can sort of interactively add all these segments. So you can have chromosomes, heat maps, links, different types of links. I think that's right. Okay, so you have each of these features, right? So you add them one by one, and then you click generate plot, or you could generate. Let's see if it gives me an error. Didn't give me an error. Okay, and you build a plot over to the right. What is the settings? Oh, you can change the title. It's very interactive, and I haven't used it enough to get really good results. But let's try to add a heat map, I suppose. Heat map would be gene expression. Now, what's interesting to me is, I guess we would add it by sample, I suppose. I don't know. Oh. And then regenerate the plot. Okay, and so that didn't turn out too well, but you can always change the circle radius to get it better. So you see how the interactivity comes into play here. It didn't work. Oh, generate. Yeah, so you would move the different circles around. Let's try to get some link data. Colors, use that. I did set the colors. Okay, well, not this is won't be the win any awards, but I think you get the idea. So if you want to use your own data, you would go back to the input data and untoggle the use package built-in data set and basically pick your project, pick your folder, and then select the file. So great question, Prithvi. So I think we should follow up. Can we actually get this working with GWAS data? I think that would be fantastic. So any questions about that? About
What does it mean by link busy?
I don't know. That's a good question. It's following the vignette. So I tried to follow it when I was preparing, but it's still a bit confusing for me. But I really do like the interactivity of this. Kritika.
Yeah, so when we are giving it our own data, what are the columns that it expects? Any idea?
It's going to let's go back to the references. It wants things in a certain format for sure. Actually, I don't know what this is. I guess these are just when these are papers that feature Circos plots. User guide. Okay. So here, let me put this in the chat. Okay, so this is a vignette. So if you guys are familiar with that, I tried to follow this vignette, but using the Shiny app, and I didn't do so well, but this really tells you how each of the data sets, but to answer basically what using the example data set would be. But to answer your question, it should be in here. I'm not sure how we need to format it.
Two more questions to piggyback on that one. I see that at least in the example that you showed, or like the code that you just showed, it was AG19. Any idea if this is restrained to AG19 build only, or can this also work with AG38? Or is there any such concern that it will not work with any other genomic build for that matter?
I'm not sure. I think 38 would be, for the most part, compatible with HT19, but we should probably double check on that. I mean, as far as, in this case, I think just the chromosome naming, you would have to be careful with.
And is there any idea if there's any restriction with respect to what file size can this
Good question.
use?
I don't know. I can follow up on that as well.
Okay.
But yeah, a lot of potential there. And we need people to try it out in the community. Any other question? Any other feedback from anyone on the call? Is there any, so we have some time left. Is there anything else you want to see or discuss?
We do open the floor for all questions too. So if there's something other than what was presented on today that you have a question about, feel free to ask away. We have other platform reps on the line.
Yeah, I just had a wrapping up slide. So apps are good. They provide value and they're ease of use. As long as you read the manual, especially for OmicsCircos. Scalability and learning enablement. So we talked about STAAR Pipeline, MetaCyto, OmicsCircos. We think that they significantly increase the capabilities of Biodata Catalyst. So in the future, we'll continue to release innovative apps. And we just need to hear from you about what you want to use. Of course, you can come to office hours, and if there's any app on GitHub that you want to try, we can really get that. We can work with you to get it set up. So any call to action. What are your pain points? How can apps make your workflow better, scalable or shareable? And a question I don't know the answer to, can we integrate genomics and cytometry data? Is that possible? So I see a point from Prithvi that, so by the app builder, do you mean the person who sort of wrapped it in CWL or got it working on the platform, or do you think like the actual software developer? Just I want to clarify that. Regardless, I think that's important feedback.
All right. Well, thank you, everybody. We do have about 10 minutes left. So again, discussion and question time. If there's any questions you have, Prithvi, you did have a good point in the chat. Be better if we could bring someone who has made the AppStava training on the app with data and answer related questions. So we definitely wanted today to be just an introduction, and thank you, Dave, for that. And maybe in a future hours, we can bring somebody like that on. I think that'd be a really valuable session. So again, about 10 minutes left for discussion and questions. Of course, you can submit questions to our help desk, but we have 10 minutes now. If anybody wants to raise anything up, again, it does not have to be about today's presentation. It can be about anything in general.
Can be about anything. And I recognize some of the names on the call. So, if there's any project you're working on, you say, I really need this particular app or this particular data set. I mean, let's use the time to discuss that.
Hello.
Hi.
Okay. Yeah. I want to ask the question. Using the BDC platform, we can actually combine two parts of chromosome, which are actually DCF file, and convert it to GDS file. So can we combine all the chromosome at once and convert it to GDS file?
So I think you have a better scalability if you keep them separate. So generally, at least with the Genesis flow, it was you would take the different you would combine chromosome 1 with all your other chromosome 1s and then convert to GDS. Is that so?
Yeah,
what are you?
The normal procedure. Yeah, we can combine different parts of the same chromosome and convert it to GDS. But if we want to convert all the chromosome at the same time to GDS file, is it possible in BDC platform?
So do you mean like convert, like let's say you had two studies and they each have all the chromosomes, right? You would want to convert them all to GDS first and then combine?
Yeah,
Is
that can
that-
also happen, no problem.
Yeah, I don't know. That's a great question. The way it was implemented was to do the BCF tools merge. But it seems to me like you could also, like there has to be a tool that would allow you to combine GDS files essentially, right? Like that's got to be
Okay.
performant. I don't know the answer, so I'm just kind of looking around. I'm not sure why it was decided to use BCF tools. But you know, Google is our friend here. So we have, looks like there's a function to merge multiple seq array GDS files. It's kind of one of those things like, I think the method you're describing makes more sense to me, but a lot of times it's really the first step in a GWAS. So people usually do it once and they kind of don't think about it anymore. So maybe that's why people aren't trying different methods. Thank you for the question. I think that's important. Important, and I was literally thinking about this week, because emerging can take some time, several hours, especially for the large chromosomes.
Yeah, but I actually tried which you have just shown, but when we are actually filtering using the same seq array, there is some kind of problem actually. It did not work properly. When I am applying seq array for a single chromosome, which is converted to GDS file, it is working perfectly. But when if I combine two GDS files and then apply seq array to filtration for further analysis, it was not actually properly working, which I can recall. So that's why I actually asked you.
How did you merge? What did you use the seq array seq merge? How did you merge it?
I have to maybe go again. So it would be better, I think, if I contact you later.
Okay, sure. Yeah, absolutely. I come to office hours, by the way.
Yeah. Okay.
So I heard crickets when we were talking about flow cytometry. Is there anyone that wants to champion flow cytometry or talk about how?
Yeah, it would be great if someone can tell about mass cytometry and flow cytometry. I have no idea about that.
Yeah, so that's what we're trying to get the community into a place where, you know, we have experts, you know, in different areas that can sort of talk. And I do want to mention community hours for next month. It's going to be excellent. I've been working with Andrew Blair, who's one of the PCGC fellows. There's 4 fellows on this cohort. They're accepting applications for another cohort of fellows. But he's going to really talk about some interesting things. He's very knowledgeable.
Yeah, the description is 2 slides over, I think, for those of
at the end or at the beginning.
At the end. I think if you just go 2 slides forward, it should be there. Just if anyone's still.
Oh, cool. Yeah. Great. And next time we'll just ask him to define what CHD means in his abstract. So it's related to, if you're fans of Bioconductor, part of what he's working on is creating summarized experiment objects for these results of massively parallel reporter assay and other assays, but also creating what's called, I don't want to steal his thunder, but he has a Shiny app, which we're going to host on Biodata Catalyst. It's also hosted by other places called IC. Time do I have? I don't think, I'm trying to find the link for it. This is just a preview for next month. I think this is a valid thing. So interactive summarize experiment explorer. Flow cell data, Joe. Joe's question is, I missed if MetaCyto works for flow cell data. What type of flow cell data? Do you have any more information? I don't. I may be out of date. When I think of flow cell, I'm thinking of sequencing. But
oh yeah, I do not know off the top of my head. It's a flow cell sorter. Other people will be working with it. Yeah, I'll have to get back to you guys on that.
yeah, we'd like to hear it, and we like to sort of build out some more use cases that are more tangible. So we'd like to hear from you. And if you have access to any data, doesn't have to be something that you want to,
Yeah, that's
share.
a good, yeah.
I can show you how to upload it, how to use the pack. Like we do that a lot where we work
Sure.
with users.
Well, it's still in the early goings here, but yeah.
Well, thank you again for everybody who joined us today. About 1 minute left, so if you have a really quick question, feel free to ask. Otherwise, watch your emails for today's recording, the slides, notes, and hopefully some answers to some of the questions, the really good questions that people had today. So all that will be made available soon, and please get in touch with us if you have additional questions. We look forward to helping you, and if you want to get started on your research, or if you have questions about your research, get in touch. We're happy to help. So I think I'll end the meeting. Hope everybody has a really great afternoon, and we will see you in June. Thanks, Dave. Thanks, everybody, for joining.